Welcome to today's video on spinal cord injury research, where we delve into groundbreaking studies and inspiring advances in medical science. Today, we focus on a study targeting microglia to improve autonomic outcomes after spinal cord injuries. Let's begin with understanding the challenges posed by autonomic central nervous system dysfunction following spinal cord injury, SCI. Spinal cord injuries can profoundly impact the body's autonomic functions, which are critical for maintaining homeostasis. These functions include heart rate, blood pressure, digestion, temperature regulation, and bladder and bowel control. When the spinal cord is injured, communication between the brain and various parts of the body can be severely disrupted. This leads to autonomic dysreflexia, a life-threatening complication that requires immediate attention. Autonomic dysreflexia occurs when there is a disconnection between the brain and the sympathetic nervous system below the injury. This can cause an exaggerated response to stimuli, leading to dangerously high blood pressure, severe headaches, sweating, and skin flushing. Triggers can be as minor as a full bladder, tight clothing, or even a wrinkle in the bedding. Autonomic dysreflexia usually affects injuries to the cord that are above the mid-body level. Suffering individuals need to be ready to immediately treat the condition, as it can very quickly become life-threatening. In addition to autonomic dysreflexia, individuals with SCI may experience other related secondary conditions such as orthostatic hypertension. A sudden blood pressure drops when standing, causing dizziness or fainting, as well as cardiac arrhythmias, irregular heartbeats, including slow or fast heart rates, thermoregulation issues, that is, difficulty regulating body temperature, leading to abnormal sweating or chills, bladder dysfunction. There are many problems with bladder control, resulting in urinary retention, incontinence, reoccurring UTIs, and high risk of kidney disease. And bowel dysfunction, damage to the nervous system, can result in lifelong challenges with bowel control, constipation, incontinence, gastrointestinal issues, and digestive problems such as bloating and acid reflux. The effects of these secondary conditions can be lifelong and debilitating. Beyond the immediate physical symptoms, chronic autonomic dysfunction can lead to long-term health issues such as cardiovascular disease, impaired immune response, and reduced quality of life. Immediately after a spinal cord injury, the affected area undergoes a complex series of events. The initial impact causes direct damage to the neurons and glial cells, leading to cell death and disruption of neural pathways. This acute phase is characterized by inflammation, swelling, and the release of harmful chemicals that exacerbate the damage. Microglia, the central nervous system's resident immune cells, play a pivotal role in this response. They rapidly migrate to the injury site, where they help clear debris and release signals that can both protect and further harm the neural tissue. As the acute phase transitions to the chronic phase, the body attempts to repair itself, but this process often leads to the formation of scar tissue. This scar tissue, while stabilizing the injury site, creates a barrier that prevents the regrowth of axons and hinders neural regeneration. This scar tissue formation, known as glial scarring, is a major obstacle to recovery. Additionally, maladaptive plasticity can occur where the nervous system reorganizes itself in a way that leads to chronic pain and other dysfunctions. This reorganization is partly driven by the ongoing activity of microglia, which can perpetuate a state of heightened sensitivity and inflammation. Understanding the dual nature of microglia's role, both protective and potentially harmful, has led researchers to explore ways to modulate their activity. The goal is to enhance their beneficial effects while minimizing the negative outcomes. The study we're focusing on today, published in the journal Science Translational Medicine, investigates a promising approach to modulate microglial activity to prevent these adverse outcomes. Conducted by Dr. Philip Popovich and his team at The Ohio State University, the study identifies a specific drug target that could help manage microglial behavior to enhance recovery and minimize complications. The researchers targeted a protein known as colony-stimulating factor 1 receptor, CSF1R, which is crucial for the survival and function of microglia. By inhibiting this receptor, the scientists aim to reduce the excessive and prolonged activity of microglia after spinal cord injury. In their experiments, the team used a drug called PLX 5622 to inhibit CSF1 receptor. This drug effectively reduces the number of active microglia at the injury site without completely eliminating them, allowing for a more controlled and beneficial immune response. The results were remarkable. In animal models, treatment with the drug led to significant improvements in several areas. Reduced scar tissue formation, the inhibition of CSF1R led to a decrease in the formation of glial scars, which are major barriers to neural regeneration. Improved neural regeneration. With less scar tissue, there was an increase in axonal regrowth, facilitating better communication pathways within the spinal cord. Decreased chronic inflammation. The controlled activity of microglia reduced chronic inflammation, a key contributor to long-term complications such as chronic pain. Enhanced functional recovery. Animals treated with PLX 5622 showed better motor function recovery compared to those that did not receive the treatment. One of the most exciting aspects of this study is its potential application to human spinal cord injuries. While the research is still in its early stages, these findings provide a strong foundation for future clinical trials. The hope is that by targeting microglia with drugs like PLX 5622, we can significantly improve recovery outcomes and quality of life for individuals with spinal cord injuries. Researchers are now focused on translating these findings into human studies with the aim of developing therapies that can be used soon after injury to maximize the benefits. The future of spinal cord injury treatment looks promising, particularly with the advances in microglia-based therapies. Building on the foundational work by Dr. Filip Popovich, one of the key areas of focus is the development of next-generation drugs that can precisely target microglial receptors. By refining molecules like PLX 5622, scientists hope to achieve even better control over microglial behavior, reducing inflammation and promoting neural regeneration without the adverse effects seen in earlier treatments. Furthermore, the combination of microglia-targeted therapies with other emerging treatments holds great promise. Researchers are investigating how these therapies can be integrated with CRISPR bioengineered scaffolds, stem cell therapy, and neuroprosthetics to create a synergistic effect, enhancing overall recovery. The future of spinal cord injury treatment is bright, with multidisciplinary approaches offering new avenues for recovery. Collaborative efforts between neuroscientists, engineers, clinicians, and patients are driving this progress forward. Each discovery brings us closer to a world where spinal cord injuries are not a life sentence, but a challenge that can be overcome. Understanding microglia and their role in spinal cord injuries is now one of many tools redefining SCI outcomes. The combination of microglia-based therapies with bioengineered scaffolds, stem cell therapy, gene therapy, neuroprosthetics, and advanced rehabilitation techniques offers a comprehensive approach to recovery. Through continued research, innovation, and perseverance, we can look forward to a future where recovery is not just possible but probable. Thank you for joining us on this journey through the cutting-edge research and emerging therapies in spinal cord injury treatment. Stay tuned for more updates and breakthroughs in this exciting field. Together we can inspire hope and make a difference.